Today I'm going to talk about the border between random evolution and intelligent design. And I'm going to start with a review of my talk from last night, just in case anybody was not around or has slipped your mind. In The Origin of Species, Charles Darwin himself wrote that if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down, adding, but I can find out no such case. In this, Darwin was insisting that evolution had to be gradual in tiny steps, improving life in very small amounts over a long period of time. And that is the difficulty that I focused on in my first book, Darwin's Black Box. It turns out that many biological systems certainly look like they cannot be formed in the gradual manner that Darwin insisted upon. And some were recognized immediately when he wrote his book, such as the eye and other systems. But the problem has become particularly acute now that the molecular and cellular basis of life has been discovered. And the problem of systems that can't be produced by in a gradual manner, I called irreducible complexity. And irreducible complexity is a fancy phrase, but it stands for a simple idea. Many machines, many systems depend on multiple parts interacting with each other in order to perform their function. And you can see that in even very ordinary objects from our everyday life, such as this mousetrap. The mousetrap has a number of parts: the wooden platform, a spring, a metal hammer, and other metal pieces as well. And even very tiny features of the system are critically important. For example, the holding bar, which stabilizes the hammer when it's pushed over here, has this little curl on the end to attach to this staple which holds it in place. If that little curl wasn't there, the whole system would be useless. So many systems need multiple parts to function, and it's very difficult to see how something like this could be made by something like a gradual Darwinian evolutionary process. For example, if you wanted to make a mousetrap, how would you start? What would you start with? Could you start with just the wooden platform and hope to catch mice inefficiently? Maybe when they ran, they might trip on the wooden platform and fall over. And then you might add this holding bar, and when they tripped, they would be impaled upon the holding bar. Well, no, that's silly. You have to have the whole thing pretty much in place before it works at all. So these irreducibly complex systems are big challenges to Darwin's theory. So the question is, are there any such things in life, especially at the foundation of life, the molecular and cellular foundation of life, that has been uncovered by science, especially in the past 75 years or so? And the answer is yes, it's all over the place. A major discovery of modern biology is that life is run by machines, literally molecular machines in the cell. And last night I spoke about the bacterial flagellum, which you know is like an outboard motor that bacteria use to swim. And it has multiple parts. It's got, there we go. It's got a propeller, and it's got a universal joint and drive shaft, and many, many other parts. So it's a big challenge to Darwin's theory. And in fact, if you look in science journals, in books, in all of the scientific literature, there is no one who has even attempted to explain how a Darwinian process could make such a machine. And irreducible complexity, as I said last night, has two important features. It is a block to Darwinian explanations, and it is a strong pointer to purposeful, intelligent design. Because we recognize intelligent design when we see a purposeful arrangement of parts. When a number of parts are put together so that they do something, so that they have a function, we always recognize the activity of a mind. And I showed the following cartoon last night to get that point across. We saw some pieces of wood and a vine and in a completely natural setting, but everybody who looks at this cartoon knew that this system was designed because we recognize the purposeful arrangement of parts. That's how we tell that something was designed. There is no other way, by the purposeful arrangement of parts. Okay, so that's what I said last night. But a question immediately arises if you're a person like myself. We see in life, particularly in cell and molecular biology, that there are many things that cannot be explained by Darwin's theory and were required to be purposely designed. And yet, not everything needs to have been purposely designed. You know, maybe the exact shape of my nose, you know, it might have been different, you know, that's okay. We know that in our everyday world, antibiotic resistance can occur. So some things can occur without purposeful design. So a question I wanted to ask was, what is the rough border? What is the limit? What is the edge of random evolution and intelligent design? What are natural processes capable of explaining, but what required purposeful direction, design, in order for it to occur? And that's what I'm going to be talking about this morning. And to begin with, I want to make some distinctions about evolution that are really critical to understanding the argument. And that is that Darwinism, Darwin's theory, is a multi-part theory. It's got several different ideas, several different concepts all bound up in it. But some parts of it might be wrong, or might be right, and others might be wrong. And one of the major concepts of Darwin's theory is the idea of common descent, that organisms alive today are descended from organisms that were alive in the past. And I think that's an interesting idea. It's an interesting idea about natural history, but ultimately it is trivial because it doesn't say where the ancestral organisms came from. It doesn't say how they changed into modern organisms. It just says there were these animals in the past and they gave rise to these animals in the present, but doesn't say how. So essentially it's interesting, but it really doesn't have much philosophical or even scientific substance to it. Another part of Darwin's theory is the idea of natural selection, that an organism which is somehow a little bit better fitted to its environment will survive better. And again, that's interesting, but really trivial, because who would deny that an organism that was better fit to its environment would have a better chance to survive? You know, that's interesting, but it's not really a big surprise. I think the critical part of Darwin's theory is the idea that random changes, random variation, random mutation can supply the variation that will build intricate, interwoven biological systems. And that has been the criticism of Darwin's theory from its inception, and it continues to be today. The critical claim of Darwinism is the sufficiency of random mutation, and that's where it is by far the weakest. And that is where intelligent design disagrees with Darwin's theory. The other ones are interesting to be debated in their own right, but the critical part is random mutation. Okay, so why should we think there is a limit to evolution, to Darwinian evolution? Why don't I think that it can explain everything? Well, one thing is that very, very complex structures, functional structures, have been discovered at the foundation of life. This is actually a computer model of a psyllium, which I showed slides of a psyllium last night. But this emphasizes all these little dots, these blue dots, dark blue, this green, this yellow. They're all themselves very complex protein parts. And yet, so I just wanted to show this to show you the purposeful arrangement of parts in this structure. But more than that, I wanted to show you where this picture was published. It was published in a journal called Nanotechnology. Nanotechnology. You know, Mackenzie has a nanotechnology program where very smart scientists are trying to make functional small systems. But we have these things. We have incredible nanotechnology in our cells already. So that's one reason why we should think there's an edge to evolution, because in fact life has turned out to be much more complex than anyone in Darwin's day ever imagined. Another reason is this. Here's a kind of a summary of Darwin's theory, and Dr. Miller showed something like this on a slide in his talk. This is supposed to be a measure of the fitness of an organism, and these are different genes. You know, this is all very theoretical, and this is supposed to be the fitness of an organism. And the idea in Darwin's theory is that if an animal has a fitness like this, and a change comes along, a mutation, to make it a little bit more fit, then it will survive better. And another mutation, it'll survive better. And another, until it's as good as it can be. Well, that's great, and that's a good idea. That was a good idea of Darwin's. But it turns out that that's just an idealized fitness landscape. This is what you would expect a realistic fitness landscape to look like, that there are many different parts. And so you can improve one aspect of an organism or one aspect of a system, but that doesn't relate to other aspects of the system. And what's more, this is the landscape here is incoherent. incoherent. I think that's incoherent. And by incoherent, I mean that rising up to the top of one peak has nothing to do with other peaks, whether it will rise on another peak. There's no way for those peaks to relate to each other. Each one rises on its own. So that's a second reason to think that Darwin's theory has limits. A third reason is that current science hasn't any clue, doesn't have any idea how such things as the cilium or flagellum might be put together. And if you look in the science literature, it's actually admitted in a low voice, kind of a whisper. For example, a couple years ago in the journal Nature, which is the leading science journal in the world, a man named Philip Ball wrote a comment in which he said, in short, the current picture of how and where evolution operates and how this shapes genomes is something of a mess. Barely a whisper of this vibrant debate reaches the public. You bet it doesn't reach the public. Criticisms of evolution and acknowledgments that people don't know how evolution, Darwinian evolution could produce such things is kept very much within the scientific community. And anybody who mentions it to the public, well, they will pay a price. Okay, so that's why I think there are limits to Darwin's theory of evolution. So how can we go about finding out what those limits are? What can random mutation and natural selection do without guidance? It can do some things, and what is required, what requires intelligent design? So what we want is, what is the best evidence, the best scientific evidence of what Darwinian processes can actually do? And I argue that the best evidence we have to assess the abilities of Darwinian processes comes from studies of malaria, both in genetic changes of humans and in the parasite Plasmodium falciparum itself. And the reason that is the best studies is that there are many detailed genetic studies on malaria because we're interested in our own health. So a lot of studies have been done, and the population sizes involved. There are many humans that are exposed to malaria. There are billions of humans who have been exposed to malaria, and there are uncounted trillions of malarial organisms that have existed. So we have huge population sizes of both humans and malaria. Here's a little figure showing the beginnings of the infection of a human by malaria. Malaria is carried by the mosquito. The malaria parasite is just a single-celled organism, and the mosquito doesn't like this either. It gets infected and is harmed by the organism. But when an infected mosquito bites a human, the malarial parasite enters and goes into the liver, where it reproduces for a few weeks and then comes out, and one malarial parasite, see that little bump there on this red blood cell, this is supposed to be a red blood cell, that one malarial parasite enters into a red blood cell and it eats the hemoglobin inside the red blood cell. It's like a vampire. It eats the hemoglobin in our red blood cell. And it uses those materials and the energy to make about 20 copies of itself. And when it makes 20 copies, now all of 20 of them burst out of the red blood cell, which is now destroyed, and each one attaches to another red blood cell and enters and makes 20 copies. So in the second round, 400 organisms burst out. And in the next round, 8,000, and so on. And it increases exponentially until it eats most of a human's blood, or very much of it. And that's what can kill a person. Well, in the 1940s, a drug was discovered, chloroquine, which could cure malaria, would kill the malarial parasite. And it was really a wonder drug. It was cheap, and it didn't have many side effects, and it was very effective. And there was much talk about the elimination of malaria, just as some other infectious diseases have been eliminated. But after about a decade or two, some malaria seemed to be resistant to chloroquine. It took more and more chloroquine to kill them. And pretty soon, some strains of malaria could not be killed by chloroquine. It became very resistant. Let's see where I am here. I want to skip over this. And this article in the early 2000s from Science magazine shows the end result, a requiem for chloroquine. It had completely lost its effectiveness to fight malaria. Well, that's interesting, and people have searched for other drugs, but it turns out that most drugs against malaria are very ineffective, and resistance to the newer drugs is developed very, very quickly. For example, let me go back a slide. Whoa, I don't want you to read that in advance here. Okay, so this is the frequency of development of resistance to malaria. And there's a drug called atovaquone, which has some anti-malarial activity. But it turns out that resistance to atovaquone arises in every third patient to which it is given. So doctors in a clinic, if there are 100 patients, they could give atovaquone to everybody, and 30 of them, roughly 30 of the patients, would spontaneously would not be cured by the atovaquone. There would be malaria in those patients that had a mutation that could resist the drug. On the other hand, if you compare it to chloroquine, resistance to chloroquine arises in about every billionth patient, about every 1 in 10 to the 20th cells. So that was the major point that I investigated or wrote about in my second book, The Edge of Evolution. And in it, I said, well, a likely explanation was given by a couple of researchers about 5 years before I published my book. And the explanation is really a simple one. If you think about a lottery, say, you buy a ticket and you can win some money. Well, suppose you had to pick a number between 1 and 100. What are the odds that you'll get the correct number? Well, it's 1 in 100 to get the number, say, 22. All right, that's fine. Well, what if you have to match 2 numbers between 1 and 100? What are the odds that you'll match 2? Is it 1 in 101? 1 in 102? 1 in 200? No, of course, you have to multiply the odds together. It's 1 in 100 times 1 in 100, which is 1 in 10,000. And if you have to match 3 numbers, that's 1 in a million. So the odds against winning go up exponentially with the number of numbers that you have to match. And I wrote in the book that if the malarial parasite has to have not just 1 mutation to resist chloroquine, but has to have 2 mutations, then since the genome, since the number of units in DNA in the malarial parasite is about 100 million, you'd expect resistance to be about 100 million times more rare for a drug that needs 2 mutations versus one that needs just 1 mutation. And some work had been published showing that 2 mutations commonly occurred in a certain protein in chloroquine-resistant malaria. And a little bit later, about 5 years after my book came out, some researchers showed that, in fact, 2 mutations were needed for any anti-chloroquine activity, for any resistance to occur in malaria. So the point is that if you need just one change, one little change, well, Darwinian processes work fine. You know, just by chance you can get one change, and if that does something helpful, well, that can be selected. Okay, that's medically important, it can be biologically important, but if you need 2 changes, then it becomes very, very, very difficult to get the 2 coordinated changes together, because the odds, the improbability, increases exponentially with each separate change that you need. And malarial parasite was able to find the 2 necessary mutations only because there are 10 to the 20th malarial parasites, and that is what? It's a 100 million trillion organisms. There are that many malarial parasites on Earth every year. Now, humans, we only have a couple billion or so. So it would take humans to come up with 2 coordinated changes, billions of years. It would effectively be impossible for an organism like us that has a smaller population size. Malaria could do it only because it has astronomical numbers of cells. Okay, so now if you think about it, you ask yourselves, well, what did Darwinian processes do in trillions upon trillions upon trillions of chances? What did it do? And the answer is, as I showed, it made 2 crummy amino acid changes in a protein that already existed in the malarial parasite. It made 2 lousy, what's the best Portuguese word for that? Very small changes in a pre-existing protein. And yet, if you think back to the psyllium, this isn't even, this isn't even one little blue dot from that machine, the picture of the psyllium machine that I showed earlier. So, where am I here? Whoop, I'm sorry, going the wrong way. So the results here, this is not an argument that Darwinism cannot make complex functional systems. This is an observation that it does not. I'm not arguing. Nature could have done anything it wanted to, and the only thing it could do in 10 to the 20th power attempts was to get 2 amino acid changes. That's an observation. It's not an argument. And it's interesting to note that sometimes people say that Darwin's theory is not the only idea in evolutionary biology today. Why, we've got self-organization, we've got all sorts of different things. But in nature, nobody was telling the malaria what to come up with. If it could self-organize, if it could self-engineer and come up with a better solution to the problem of chloroquine, it was free to do so. But in 10 to the 20th attempts, the only thing it could come up with was small microevolutionary amino acid changes. So no unintelligent process helped malaria very much, including Darwinism and, in fact, the other newer mechanisms that people tout these days were not even seen. So Darwin's old idea did better than the newer ideas that people have come up with. Okay, so how does this inform our idea about the limits of Darwin's theory? Well, in a book 12 or so years ago, the intelligent design proponent Jerry Coyne, that's a joke. No, the Darwinian biologist Jerry Coyne and his student Alan Orr wrote a book, Speciation, and they wrote that the goal of theory is to determine not just whether a phenomenon is theoretically possible, but whether it is biologically reasonable. That is, whether it occurs with significant frequency under conditions that are likely to occur in nature. Okay, so what types of things will occur within the limits that we see in the case of the malaria parasite that would require, say, 2 amino acid changes or more? And it turns out that just to get 2 proteins to stick to each other, to bind to each other, as they routinely do in a cell, requires multiple complementary amino acid positions in the 2 proteins. So that each of these little illustrations here is supposed to represent a feature of a protein, such as a hydrophobic feature or a positive charge and a negative charge and other such things. So you would expect to need multiple changes to get 2 proteins to stick together that didn't stick together before. And so that would be a problem at least of the order of developing resistance to chloroquine. And in my book, I argued that, in fact, to get 2 proteins, or 2 new protein-protein binding sites to develop by random changes and natural selection would require 10 to the 40th organisms. 10 to the 40th power organisms. And it turns out that is slightly more organisms than have ever existed in the entire history of the world, including microorganisms. So essentially, 2 protein-protein binding sites is the limit of evolution, of Darwinian evolution. And down here, I have little dots showing that in how many numbers of organisms new binding sites have developed. And we have 1 for sickle hemoglobin and none in the case of malaria and many other examples in the literature. So this is the limit of evolution, and this is what we have seen through observation, and this is what we need. So, 2 protein-protein binding sites, and yet the cell is filled with machines like the cilium, in which these different colors, structures are held together by different protein-protein binding sites. So clearly this is very far beyond the edge of evolution. Well, most people don't care about biochemistry, I'm afraid. They care about animals and plants and pretty little rabbits and things like that. So in organismal terms, what is the likely edge of evolution? Here's an organism, the sea urchin, which has been studied very much by developmental biologists, looking to see what at the molecular level is necessary for its tissues to develop correctly. And here's a diagram of the various proteins involved. Each one of these little things here is a different protein that is involved in the process. And as you can see, it looks like a complex electrical or computer diagram. And this is to make just one tissue. And I have argued, based on various pieces of evidence based on this, that you might be able to get 2 or 3 new such interactions. And advances in biology have shown that you need at least 5 or 6 changes to make a new class of organisms. That a class is like fish versus birds versus mammals. And so I wrote in the book that all of this in nature had to have been designed. The laws of nature, the constants of nature, the origin of life, cells, molecular machines, and so on. But as you get down, we're going from more general to more specific. And I think that there are accidents in biology, there are random mutations, and so on. And I think various species can arise, if you think of different species of birds, say. But somewhere between the level of class and the level of species in genera is the edge of evolution. Now, lately, I have been looking at some other results of experiments, and it turns out that much Darwinian evolution proceeds not by changing amino acids or by improving something, but by breaking old genes. And that's actually what you might expect in retrospect, because disabling a gene, breaking it, will occur at a rate hundreds of times faster than making a specific improvement in a gene, simply because there are many more ways to break a gene than to improve it. Also, very, very few of the possible accessible mutations are helpful. There were only 2 amino acid changes in one protein in malaria that could help it. But there would be mutations in every protein over the population of malaria cells in the world, but only those 2 could help. And as I mentioned before, random changes are incoherent. But let's talk about this point, that disabling a gene will occur at a rate hundreds of times faster. Here's an example of a beneficial mutation. Okay, does everybody read Batman and Robin down here? Okay, I hope so. Anyway, Batman and Robin were going to arrest the Joker, and they were driving across the bridge, but the Joker blew up the bridge. And there they go, they're diving in the water. They're not hurt. But if you think about it, This is a beneficial change for the Joker. This is a beneficial mutation to destroy this structure. If he did not destroy this structure, he would be arrested and jailed and so on. It turns out that genes are just like that. Sometimes it's literally you blow up a gene and it stops an organism from invading. For example, here are mutations in the human genome that help, that give some resistance to malaria. Let me look at one. Down at the bottom, there's a mutation called Duffy antigen. And it's a point mutation, but what it does is it turns off a gene that malaria cells recognize the protein that the gene codes for, and you bind to it and use that protein to enter into the cell. But in some populations of humans, they have disabled that gene. They have blown it up. So now the malaria can't get across the bridge into the red blood cells. So it's a pretty much the same thing as encountered here. But blowing up a bridge or blowing up a structure does not tell you how the structure was made. It's not the opposite of making the structure. Here's another example. A gene was discovered in populations of humans which helped them resist heart attacks. They could eat food that was high in fat and not have high cholesterol, not have heart attacks. What was the mutation? It was a mutation that destroyed the function of a previously existing protein. It degraded the protein, and that helped. That was a beneficial mutation. There's a mutation in horses that makes them trot in a smoother manner, which is preferred by many riders. And it was recently discovered what the mutation was. And it turns out that it was a loss of a gene. A gene no longer works, and that was beneficial. The horse was selected because of that. Recently, the genome of the polar bear was sequenced, and the sequence of the brown bear's genome was sequenced earlier, and the two were compared, and the polar bear is thought to have descended from the brown bear. Well, how do you get a polar bear from a brown bear? It turns out that one of the major differences between polar bears and brown bears is that the polar bears can eat a lot of fat, like seal blubber, and they burn that to stay warm in their cold environment. Well, the researchers looked at the genes that were most highly selected in the polar bear, and it turns out that many of them were destroyed, or they had damaging changes. So about 50% of the changes in the genes that they identified were damaging ones. So new species can arise by Darwinian processes, but the random mutations destroy information. They destroy genes. So that can lead to a little bit of variation, but it's not creating anything. Different dog breeds. In this era, the sequences of the genomes of different dog breeds can be elucidated. And I won't go through them, but most of the gene changes destroy the gene. And, I've got lots and lots of stuff, but so I wrote a review paper in 2010. where I reviewed work on bacteria in laboratory evolution experiments. And I summed up the result saying that most of these bacteria improved, but they improved by breaking genes. And so I coined a phrase called the first rule of adaptive evolution, and that is to break or blunt any functional coded element, like a gene, whose loss would yield a net fitness gain. Okay, I'm going to skip ahead here to close to the final, Oh, this is great, this is great. Oh, too bad I can't go into that. Oh, that would be great. Okay, so the long and the short of it is that the edge of evolution I now estimate to be at the level of families. Families, there is much less certainty. It's gone down from here. This is kind of like a logarithmic scale, so this narrowing of it actually represents the ability of evolution, descending from making fish versus birds to be able to make, a few changes in the Galapagos finches and other such things. So this, I think, is the edge of evolution, the family level, such as cats and dogs and bears and raccoons and seals in the mammalian order. And that level of life required purposeful, explicit information in order for it to occur. And then these biological categories could diversify by losing information, by breaking different genes. Okay, so again, I showed last night that there have been a few critical responses to my arguments, and that a good place to find my responses to those criticisms is on that website. So thanks very much for your attention.